Welcome back to the Deep Dive. Our mission today is pretty specific.
Hmm.
We're diving deep into the genetics of human disease. We want to cut through some common misunderstandings.
Right.
And zero in on, well, the most common pattern of single-gene inheritance.
Hmm.
We're talking about autosomal dominant disorders, or ADDs.
ADDs, it's a big topic.
It really is.
Yeah.
And, you know, when you hear genetic disease, I think maybe people often think of things that are incredibly rare, almost like textbook examples only.
Right, something obscure.
Exactly.
Yeah.
But the stats we pulled from our sources, they paint a very different picture, a much larger one, really. The lifetime frequency estimate for genetic diseases, it's something like 670 per 1,000 people. Staggering.
It is staggering. And that figure covers everything, not just late onset conditions. And the scale of underlying genetic issues is just profound. If you look at, say, early miscarriages, spontaneous abortions, research pretty consistently shows that a full 50%, half of them, have a clear chromosomal abnormality. So yeah, genetics isn't some niche area of medicine. It's really the bedrock.
Okay, so let's unpack this then. The core ideas behind these single gene disorders, the ones we call Mendelian disorders. We probably need a map first, right?
Definitely. So when we classify genetic disorders, we usually put them into three big buckets. First, you've got the chromosomal disorders, those big structural issues or number problems we just mentioned.
Okay.
Second, there are the complex or multifactorial disorders. Think chronic diseases, heart disease, diabetes, high blood pressure, where lots of genes interact with environment, lifestyle, all that.
Got it. The really common stuff.
Yeah, exactly. But our focus today, it's squarely on that third group. The single gene mutations, the classic Mendelian disorders.
Right.
These are caused by mutations in just one gene, and crucially, they usually have pretty big effects. They're highly penetrant, we say, and they follow those predictable inheritance patterns Mendel first worked out ages ago.
And within that group, we're zeroing in on autosomal dominant disorders. So what's the absolute must-have feature for a disease to be called autosomal dominant?
The definition is simple but powerful. The disorder shows up in the heterozygous state. That's the key.
Heterozygous meaning?
Meaning you only need one copy of the faulty gene to have the disease. You get one normal version, one normal allele from one parent, and one mutated allele from the other parent. And that single bad copy is enough. It overrides the good one, essentially.
Okay, so one bad copy does the damage. That makes the transmission rules pretty clear, but wow, impactful for families.
Oh, absolutely. Because the gene is on an autosome, not a sex chromosome, it affects males and females equally. Both men and women can pass it on.
Makes sense.
But the really critical number, it's the recurrence risk. When someone who has the condition, a heterozygote, has a child with someone who doesn't have it, every single child they conceive has a 1 in 2 chance, 50%.
50%. Every single time. It's not like it gets diluted or changes over generations.
Nope. A straight coin flip with every pregnancy. Imagine the weight of that.
Yeah, 50%. It completely changes how you think about planning a family, I'd imagine.
It really does. And that 50% rule leads us right to a super important question. Why? Why is one bad copy sometimes enough to cause a dominant disease when, for recessive disorders, you need 2 bad copies?
Right, because we usually learn that autosomal recessive disorders often involve problems with enzymes. Right.
Exactly. Enzyme proteins. And typically, if you lose 50% of an enzyme's activity because one gene copy is bad, the other 50% from the good copy is often, well, enough. There's a built-in safe amount of margin usually.
We can cope with half capacity.
We often can, yeah. But autosomal dominant disorders, they break that safety margin rule. And the reason is what kinds of proteins are usually involved.
Oh, okay, so it's not usually enzymes and ADDs.
Not typically. Instead, ADDs frequently involve mutations in receptor proteins. Think of them like the cell's antennas controlling activity or structural proteins.
The actual building blocks.
Precisely. The components that give tissues their shape and strength, like collagen. So if you lose half your LDL receptors, which clear cholesterol, or if the structural proteins you do make are messed up by the bad copy, that 50% loss, or that disruption, is enough to cause the full disease. There's no buffer.
Got it. That distinction is key. Okay, so let's dive deeper into the characteristics.
Right.
Beyond that basic 50% risk, ADDs seem kind of notorious for being, well, tricky to follow in families. They don't always make a nice clean line on the family tree. Why is that?
Yeah, that unpredictability is where the genetics really hits the clinic floor. There are a few crucial things we need to discuss. One is de novo mutations, new mutations, and then there are these two concepts that often get mixed up, penetrance and expressivity.
Okay, let's start with the new mutations. De novo, that means new, right?
Exactly. So for some dominant disorders, especially those that are really severe and might prevent someone from having children, easily reducing their reproductive fitness, we say a good chunk of the cases we see actually started fresh.
Meaning neither parent had it.
Correct. The mutation happens spontaneously, just once, in either the egg or the sperm cell that formed the affected individual.
So in those cases, the parents are fine, and importantly, the patient's brothers or sisters aren't at increased risk either.
That's right. The family history might be completely clear, but it's still fundamentally a dominant disorder in that affected person.
And there's this really fascinating detail from the research about paternal age, isn't there?
Yes, it's quite compelling. There's a strong statistical link suggesting that many of these new dominant mutations seem to pop up more often in the germ cells, the sperm of relatively older fathers.
Why would that be?
Well, think about the biology. Women are born with all their eggs, which just age over time, but men produce sperm continuously throughout their adult lives. That process, spermatogenesis, involves ongoing cell division, millions and millions of times.
More chances for mistakes.
Exactly. Each cell division is a chance for a replication error, a mutation to occur. Over decades, those chances accumulate. So the older the father, the higher the theoretical risk of one of those errors leading to a new dominant mutation in a sperm cell.
That makes biological sense.
Uh,
Okay, now let's tackle those two tricky modifiers. Incomplete penetrance and variable expressivity. People mix these up all the time.
all the time. They sound similar, but they mean very different things. Let's start with incomplete penetrance.
Okay.
Penetrance is about whether you show the trait at all. With incomplete penetrance, someone inherits the dominant mutant gene, but they appear completely normal, phenotypically normal, asymptomatic.
So they have the gene but none of the symptoms, like a silent carrier.
Precisely. They are a silent carrier. They carry the mutation, could pass it on, but personally show no signs of the disorder. It makes the disease look like it's skipped a generation in the family tree.
So someone could have, say, the gene for Marfan syndrome, feel totally fine their whole life, but their kid gets it really badly.
Exactly that scenario. Penetrance is often expressed mathematically as a percentage. If a disease has, say, 80% penetrance, it means 80% of people who inherit the gene will show some sign of it, but 20% won't, even though they carry the mutation.
Okay, that's incomplete penetrance. Gene present, but maybe no disease. How is that different from variable expressivity?
With variable expressivity, everyone who inherits the mutant gene does show the trait. It never skips a generation in that sense. Penetrance is 100%.
What?
But the way the trait shows up, or how severe it is, can vary enormously between different people, even in the same family.
So everyone gets it, but it looks different on different people.
Exactly. And the classic example here, the poster child really, is neurofibromatosis type 1, NF1.
Right, I've heard of that one.
Yeah. So someone with the NF1 mutation might just have a few of those light brown skin spots, the cafe au lait spots, maybe some freckling in the armpits, very mild. That's their expression of the gene.
Okay, seems manageable.
But their child, or maybe their sibling, who has the exact same FBN1 mutation,
Cool.
could have a totally different, much more severe picture. Multiple large, disfiguring skin tumors called neurofibromas, maybe serious bone problems like scoliosis, or even learning disabilities.
Wow, same gene, drastically different outcomes.
Dramatically different. The gene is expressed in all of them, but the severity, the expression varies wildly.
So how do you tell them apart in the clinic? If someone says, my uncle had the gene but was fine, is that penetrance or expressivity?
That's the tricky clinical question. If the uncle was truly examined and had absolutely zero signs, no skin spots, no eye findings on slit lamp exam, nothing, then that would point towards incomplete penetrance.
Okay.
But if on careful examination, the uncle actually did have maybe one or two tiny cafe au lait spots that everyone just ignored, or maybe some Lish nodules in the iris only seen by an ophthalmologist, then the trait was expressed just very mildly. In that case, it's variable expressivity.
So it hinges on proving the trait was truly absent versus just minimal.
Exactly. It requires careful clinical assessment.
And then there's the added layer of delayed onset.
Oh, absolutely. That's another huge factor, especially for counseling. Many very serious ADDs, Huntington's disease is the prime example, often don't show any symptoms until well into adulthood, maybe in their 30s, 40s, sometimes even later.
Which
Which means the person might have already had children, passing the gene on, before they even realized they carried it themselves. It adds another layer of complexity to family planning and genetic testing decisions.
So all these things, penetrance, expressivity, delayed onset, they lead to a big question. Why the variability? If it's the same gene mutation, why isn't the outcome the same?
Yeah, that's the million-dollar question. The sources point pretty clearly to the influence of other factors. Modifier genes, other genes in the person's genome, and environmental factors play a huge role.
Can you give an example?
Sure. Take familial hypercholesterolemia, FH, again. The main gene defect in the LDL receptor is fixed. But how high someone's cholesterol actually goes and how early they develop heart disease, that's heavily modified by things like their diet, especially fat intake, whether they smoke, other genetic predispositions to heart disease.
So the main gene mutation sets the stage, but other things influence the actual performance.
Perfectly put. The mutant gene starts the process, but the environment and the rest of your genetic background fine-tune the outcome.
Okay, this is getting really interesting. Let's move from the family patterns to the molecules. Why? How does just one bad gene copy manage to wreak so much havoc? Our sources break this down into like 3 main molecular reasons, right?
That's right. These 3 molecular mechanisms really explain why some loss-of-function mutations cause dominant disease while others cause recessive disease. Most mutations do lead to less protein or a faulty protein, but the consequence depends heavily on what that protein does.
Okay, mechanism number 1, haploinsufficiency. What does that mean?
Haploinsufficiency is, in a way, the simplest explanation. Haplo meaning half, insufficiency meaning not enough. It basically assumes that you need close to 100% of the normal protein function for health.
So 50% just isn't good enough.
Exactly. If you have a loss-of-function mutation, the one healthy allele you have can only produce 50% of the normal amount of protein. And the core idea of haploinsufficiency is that remaining 50% is simply not enough to keep things running properly. The system can't compensate.
Where do we see this most often?
We often see it in complex metabolic pathways that don't have much built-in redundancy,
Yeah.
or pathways that are under very tight control, like feedback inhibition. The classic example, again, is familial hypercholesterolemia.
FHRE.
When you
Yeah.
lose 50% of your LDL receptors, the things that pull cholesterol out of your blood, the whole system gets overwhelmed. There's just no way for the remaining 50% of receptors to handle the normal load. Cholesterol backs up dramatically in the blood. That's pure haploinsufficiency.
Okay, 50% isn't enough. Makes sense. Now, the second mechanism sounds more aggressive, the dominant negative effect.
It is more aggressive. This mechanism is different because the faulty protein produced by the mutant gene doesn't just fail to do its job. It actively interferes with the protein produced by the normal gene.
So the bad apple spoils the bunch.
Exactly. It's like sabotage. This happens mostly with structural proteins, things like collagen, keratin, components of the cytoskeleton proteins that need to assemble correctly into larger complexes or structures.
Okay, how does the sabotage work?
Well, imagine collagen. Normal collagen is a triple helix, 3 protein chains wound together perfectly. Let's say you have one mutant gene making faulty collagen chains and one normal gene making good chains. So roughly half your chains are bad, half are good.
Right.
When the cell tries to assemble the triple helix, those faulty chains often still get incorporated. But a helix containing even one bad chain might be unstable, misfolded, or just unable to function correctly. The cell might even recognize it as faulty and destroy the entire complex.
Ah, so you don't just lose the function of the bad chain, you lose the function of the good chains it got tangled up with.
Precisely. It's a dominant negative effect. Instead of just losing 50% of your functional collagen, like in haploinsufficiency, you might end up losing 75% or even more because the bad chains poison the assembly process.
That explains why defects in structural proteins often cause such severe dominant diseases, like some forms of osteogenesis imperfecta, the brittle bone disease.
Exactly. The functional output is dramatically reduced, far more than just 50%, because of this active interference. It's a much more potent mechanism than simple haploinsufficiency for these kinds of proteins.
Okay, dominant negative active sabotage. What's the third way a single bad gene causes dominant disease? Gain-of-function mutations? This sounds totally different.
It is fundamentally different. Here, the mutation doesn't cause a loss of the protein's normal job. Instead, it gives the protein a new job or makes it do its normal job in an uncontrolled, often harmful way.
So it's not about missing function. It's about gaining a wrong function.
Exactly. The mutation creates a protein that might become toxic or might signal constantly when it shouldn't, or might aggregate and clog up the cell. The protein gains a new detrimental property.
And the classic example here is
Huntington disease. Tragically classic. The mutation is an expansion of a repeating DNA sequence, CAG repeats, in the Huntington gene. This leads to an abnormally long string of glutamine amino acids in the Huntington protein.
and that makes it.
It makes the protein prone to misfolding and clumping together, forming aggregates. These aggregates are directly toxic to certain neurons in the brain. This toxicity is a new property gained by the mutant protein. It poisons the cells, leading to progressive neurodegeneration.
And because this toxic effect happens, even when the normal protein from the other allele is present, the disease is dominant.
Correct. The gain of toxic function overrides everything else.
Wow, okay. 3 distinct ways.
Yeah.
Not enough protein, haploinsufficiency. Bad protein sabotaging the good, dominant negative. Or protein gaining a toxic new function, gain of function. That really clarifies things.
Yeah. It provides a strong molecular framework for understanding why these disorders behave the way they do.
Absolutely. Let's use that framework now and look at some specific examples. How about Marfan syndrome? You mentioned it earlier. It's a classic ADD affecting connective tissue, right?
Yes, Marfan is a perfect case study. It highlights defects in structural proteins and has a really interesting dual pathogenesis that we've come to understand more recently.
Okay, so what's the gene and protein involved?
The gene is FBN1 on chromosome 15. It codes for a large glycoprotein called fibrillin-1.
Fibrillin-1. And what does that do?
Fibrillin-1 is the main component of extracellular microfibrils. Think of these microfibrils as a crucial part of the scaffolding in connective tissues. They provide the framework for elastic fibers, which are essential in tissues that need to stretch and recoil, like the aorta, ligaments, and the little fibers, the ciliary zonules, that hold the lens of the eye in place.
So it's a structural protein. Which mechanism applies here? Haploinsufficiency or dominant negative?
Yeah. It's thought to be a combination, or it can be either, depending on the specific mutation. Many mutations likely act in a dominant negative way where the abnormal fibrillin disrupts the assembly of functional microfibrils. Others might simply lead to not enough fibrillin being produced haploinsufficiency. Either way, the result is weak microfibrils.
But you said there's a dual pathogenesis, more
Yeah.
than just weak scaffolding.
Yes, and this is the really crucial insight from recent research, the part that's changing how we think about treatment. It turns out that normal fibrillin microfibrils do more than just provide structure. They also act like a storage depot for a powerful growth factor called TGF-beta, transforming growth factor beta.
Okay, TPF-beta, what does it do?
It's involved in lots of cellular processes, including growth and inflammation. Normally, the microfibrils bind to TGF-beta and keep it inactive, sequestered away.
Ah, so the scaffolding holds on to this growth factor, keeping it quiet.
Exactly. But in Marfan syndrome, with defective or insufficient fibrillin-1, the microfibrils can't hold on to TGF-beta properly. So too much active TGF-beta gets released into the tissues.
And that causes...
This excessive, uncontrolled TGF-beta signaling actually causes a lot of the damage. It promotes inflammation, it contributes to the breakdown of the tissue matrix, and it has harmful effects on the smooth muscle cells in the blood vessel walls, particularly the aorta. It's this excess signaling that's now thought to be a major driver of the dangerous aortic dilation.
Wow, so it's not just weak structure, it's also this runaway signaling molecule causing damage. That's complex.
It is. It explains why Marfan affects so many different systems, what we call pleiotropy, one gene defect, multiple downstream effects.
Let's list some of those effects. Skeletal signs are pretty striking, right?
They are. People with Marfan are often unusually tall, with disproportionately long arms, legs, fingers, and toes. That finger characteristic is called arachnodactyly spider fingers. They also tend to have very flexible, lax joints, spinal curves like scoliosis, and chest deformities, either a sunken chest, pectus excavatum, or a protruding one, pectus carinatum.
And the eyes. You mentioned the lens.
Oh. Yes, the ocular sign is highly characteristic. It's called ectopia lentis, the dislocation or subluxation, partial dislocation, of the lens in the eye. It usually happens in both eyes.
Why does the lens slip?
Because those ciliary zonules, the tiny ligaments holding the lens in place, are rich in fibrillin. When they're weak due to the faulty fibrillin, they can't hold the lens securely, so it shifts out of position. It's a major diagnostic clue.
But the really life-threatening problems are cardiovascular.
Absolutely. That's the main concern. The weakness in the connective tissue of the aorta, especially the ascending aorta, right where it leaves the heart, is the biggest danger.
What happens to the aorta?
The wall weakens and starts to stretch, leading to progressive dilation or widening of the aortic root. Histologically, we see something called cystic medianecrosis, basically. The middle layer of the aortic wall degenerates, losing its structure.
Which makes it prone to.
Prone to catastrophic failure, either severe leakage of the aortic valve, aortic incompetence, or the most feared complication, aortic dissection. That's where the inner layer of the aorta tears and blood surges into the wall itself, potentially leading to complete rupture. Aortic dissection or rupture is still the major cause of premature death in untreated Marfan syndrome.
That's terrifying. Given all this, and you mentioned lots of different mutations, is diagnosis straightforward?
It's actually quite complex because of that variability. There are over 1,000 different mutations known in the FBN1 gene. This leads to huge variation in how severe the disease is and which features are most prominent.
So how do doctors decide?
They use a specific set of diagnostic rules called the revised Ghent criteria. It's a point system based on features in different organ systems: skeletal, cardiovascular, eyes, lungs, skin, plus family history and genetic testing. You need a certain combination of major and minor criteria to confirm the diagnosis. It requires a thorough workup.
Okay, Marfan syndrome, a structural protein defect causing both weakness and signaling problems. Let's switch gears now to a different kind of ADD. What about familial hypercholesterolemia, FH? You've used it as an example of haploinsufficiency. This one's about receptors, right? And it's pretty common.
Very common, relatively speaking, for a Mendelian disorder. About 1 in 200 people are heterozygous for FH. It's a major public health issue because it dramatically increases the risk of premature heart disease.
And the core problem is high cholesterol.
Extremely high cholesterol, right from birth. Heterozygotes typically have LDL cholesterol levels, that's the bad cholesterol, that are 2 to 3 times higher than normal.
What does that lead to physically?
Over time, this excess cholesterol deposits in various tissues. You can see yellowish cholesterol deposits in tendons, especially the Achilles tendon or tendons in the hands. Those are called tendinxantomas. More dangerously, it deposits in artery walls, causing severe accelerated atherosclerosis, hardening of the arteries.
And the risk of heart attack is much higher, much earlier.
Dramatically higher. Heterozygotes have a significantly increased risk of having a heart attack, myocardial infarction or MI, in their 40s or 50s, sometimes even earlier.
And you mentioned homozygotes earlier, people who inherit 2 bad copies. How bad is it for them?
It's devastating. Homozygotes have virtually no functional LDL receptors. Their LDL cholesterol levels can be 5, 6, even 10 times normal.
Wow.
They often develop severe atherosclerosis in childhood and can suffer fatal heart attacks before the age of 20. It's a medical emergency requiring very aggressive treatment, often including things like LDL apheresis, which is like dialysis for cholesterol.
Okay, so let's revisit that LDL receptor pathway. Remind us how it's supposed to work and why the liver is so central.
Right. The liver is the main player in clearing LDL cholesterol from the blood. About 70% of LDL is removed via these specialized LDL receptors on the surface of liver cells.
How does the receptor grab the LDL?
The LDL particle has a protein on its surface called ApoB. The LDL receptor specifically recognizes and binds to ApoB. Once bound, the receptor LDL complex gets pulled into the cell.
How does it get pulled in?
It clusters in specialized patches on the cell membrane called coated pits, which then pinch off to form vesicles that go inside the cell. Inside, the vesicle fuses with a lysosome, an acidic compartment. The acidity causes the LDL particle to detach from the receptor.
And then what happens to the LDL and the receptor?
The LDL particle is broken down, releasing its cholesterol for the cell to use, and the receptor. Normally, it gets recycled back to the cell surface, ready to bind another LDL particle. It's a very efficient cycle.
But there's also that internal control system, right? The cholesterol thermostat.
Exactly. The cholesterol that's released inside the liver cell acts as a signal. High intracellular cholesterol tells the cell, okay, we have enough.
How does it send that signal?
It does 3 main things. First, it suppresses the cell's own cholesterol production by inhibiting a key enzyme called HMG-CoA reductase. Second, it tells the cell to make fewer new LDL receptors. We don't need to import so much anymore. And third, it influences the breakdown of existing receptors via that PCSK9 enzyme we'll talk more about later.
Okay, so FH messes up this elegant system. You mentioned 3 main genes are usually involved.
Yes. The lion's share of cases, maybe 80–85%, are caused by mutations directly in the LDLR gene itself, the gene coding for the LDL receptor.
So heterozygotes just make half the normal number of functional receptors.
Pretty much, that's the haploinsufficiency. With only half the receptors, LDL isn't cleared efficiently and it builds up in the blood. Plus, because less cholesterol enters the liver cells, the cells don't get that stop signal, so they may even ramp up their own cholesterol synthesis, making things worse.
What are the other causes?
A smaller percentage, maybe 5–10%, have mutations in the ApoB gene. Remember, ApoB is the protein on the LDL particle that the receptor binds to. If ApoB is mutated, the LDL particle can't dock properly with the receptor, even if the receptor itself is normal.
So the key doesn't fit the lock correctly.
Good analogy, yes. And the third cause involves the PCSK9 gene. This is fascinating. PCSK9 is an enzyme that normally helps degrade LDL receptors. If you have an activating mutation, a gain-of-function mutation in PCSK9.
It becomes overactive.
Exactly. The overactive PCSK9 destroys LDL receptors too quickly before they can be recycled effectively. So you end up with fewer receptors on the cell surface, leading to high LDL, similar to having a faulty receptor gene.
That's amazing. And I remember the source material breaking down the LDLR mutations themselves into different classes based on how they failed. Can we touch on that? It seems really illustrative.
Absolutely. It's a great way to see the different ways haploinsufficiency can manifest at the molecular level. There are 5 main classes.
Class 1.
Class M mutations are essentially null alleles. There's a complete failure to synthesize the receptor protein at all. Nothing gets made.
Class 2.
Class 2 mutations are transport defects. The receptor protein gets made in the endoplasmic reticulum, but it's misfolded or processed incorrectly, so it gets stuck. Yeah. It never makes it to the Golgi apparatus for further processing and definitely never reaches the cell surface.
So it's made but trapped inside. Class 3.
Class 3 mutations affect binding. The receptor makes it to the cell surface, but its structure is altered, so it can't bind effectively to the LDL particle. The lock is there, but the key doesn't fit.
Class 4.
Class 4 mutations affect internalization. The receptor makes it to the surface, and it can bind LDL, but it fails to cluster into those coated pits properly. So it binds the LDL, but can't bring it into the cell.
Stuck on the doorstep. And Class V.
Class V mutations are recycling defects. The receptor binds LDL, gets internalized, goes to the lysosome, but then it fails to release the LDL particle properly in the acidic environment. Because it stays stuck to the LDL, it gets degraded along with it, instead of recycling back to the surface.
Wow. So many different ways for the process to fail, all leading to the same outcome. Not enough functional receptors on the surface.
Precisely. It's a beautiful illustration of how complex protein function is, and how vulnerable it can be to single gene defects.
Okay, let's do one more case study to round this out. Ehlers-Danlos syndromes, EDS. You mentioned this involves collagen and heterogeneity. What does that mean?
Right. EDS isn't just one disease. It's actually a group of related connective tissue disorders. The common theme is defects in collagen, either the collagen proteins themselves or the enzymes needed to process and assemble collagen correctly, or sometimes other proteins in the extracellular matrix.
And it shows that link between structural proteins and dominance again.
It does, very clearly. The forms of EDS that follow an autosomal dominant pattern are typically those caused by mutations directly in the collagen genes. For example, the classical type often involves mutations in type V collagen genes, COL5A1, COL5A2.
And the really severe one.
The vascular type, which is particularly dangerous, is usually caused by mutations in the COL3A1 gene, which codes for type 3 collagen.
Why is type 3 collagen so critical?
Type 3 collagen is a major component of tissues that need to be strong but also distensible, like the walls of blood vessels, the intestines, the uterus, and the skin. Defects in type 3 collagen make these tissues incredibly fragile, leading to a high risk of spontaneous rupture of arteries, the bowel, or the uterus during pregnancy. It's a life-threatening condition, and many of these COL3A1 mutations act as dominant negatives. The faulty type 3 collagen chains mess up the structure of the entire collagen fibril.
You also mentioned there are recessive forms of EDS. How does that fit the pattern?
It fits perfectly. The rarer autosomal recessive forms of EDS usually don't involve mutations in the main structural collagen genes. Instead, they're often caused by deficiencies in enzymes needed for collagen modification.
Ah, back to the enzyme rule.
Exactly. For instance, the kyphoscoliosis type of EDS is caused by a deficiency in lysyl hydroxylase, an enzyme that modifies collagen. The dermatosparaxis type involves deficiency in an enzyme called procollagen peptidase. Because these are enzyme deficiencies, you usually need two bad copies to cause the disease, hence recessive inheritance. 50% enzyme activity is often enough.
For structural gene defect, usually dominant, enzyme defect, usually recessive, EDS shows both sides beautifully.
It really does.
Regardless of the genetic cause, what are the common physical signs people associate with EDS?
It all comes down to that faulty connective tissue. The most recognized features are unusually stretchy, hyperextensible skin, which is often soft or velvety and bruises very easily, and hypermobile joints, joints that can bend way beyond the normal range.
The double-jointed idea.
Exactly, but often to an extreme degree. This joint hypermobility can lead to frequent dislocations, subluxations, chronic pain, and early onset arthritis. There's a common belief that many professional contortionists probably have a form of EDS.
Wow. So what looks like a performance skill is actually a sign of a potentially serious underlying disorder.
In many cases, yes. That extreme flexibility comes at the cost of tissue fragility and instability. Easy bruising is also a hallmark because the walls of small blood vessels are fragile too.
Okay, that covers our case studies. Let's move into the final section, which is always the most hopeful. Translational medicine. How is understanding all these molecular details, haploinsufficiency, dominant negative gain of function, actually leading to better treatments for ADDs?
This is where the science really pays off. Knowing the precise molecular why allows for truly rational drug design. And FH, familial hypercholesterolemia, is probably the best success story here.
Because of statins.
Primarily, yes. Statins were developed based directly on understanding that LDL receptor pathway and its regulation.
Remind us how they work again. They block cholesterol production inside the cell, right? HMG-CoA reductase inhibitors.
Correct. They inhibit HMG-CoA reductase, the rate-limiting enzyme in the cell's own cholesterol synthesis pathway.
Why does blocking internal production help when the main problem is clearing external cholesterol?
It's a beautifully clever indirect mechanism. By reducing the amount of cholesterol inside the liver cell, you essentially trick the cell into thinking it's starved for cholesterol.
And that removes the brake signal.
Precisely. Remember, high intracellular cholesterol normally tells the cell to make fewer LDL receptors. When you lower intracellular cholesterol with a statin, that braking signal is removed. The cell responds by upregulating the LDLR gene. It tries to make more LDL receptors to pull in cholesterol from the outside.
So even though the person only has one good copy of the LDLR gene, the statin pushes that good copy to work harder, making more receptors than it normally would.
Exactly. You increase the number of functional receptors on the cell surface. Even if each receptor is only working at 50% capacity compared to normal, having more of them significantly boosts the overall clearance of LDL from the blood. It's a fantastic example of leveraging pathway knowledge.
And then came the PCSK9 story, which added another layer.
Yes, the PCSK9 discovery was remarkable. It started by studying people who had naturally very low LDL cholesterol. Scientists found that many of them had loss of function mutations in the PCSK9 gene.
Meaning their PCSK9 enzyme didn't work well.
Right. And remember, PCSK9's job is to tag LDL receptors for destruction. If your PCSK9 is underactive, your LDL receptors don't get destroyed as quickly. They last longer on the cell surface, recycling more times and clearing more LDL.
So finding people with naturally broken PCSK9 led to the idea of intentionally breaking it in people with high cholesterol.
Essentially, yes. It led to the rapid development of PCSK9 inhibitors. These are typically monoclonal antibodies that bind to and neutralize the PCSK9 enzyme in the bloodstream.
How does that help?
By taking PCSK9 out of action, you prevent it from binding to the LDL receptors and targeting them for degradation. The result? LDL receptors stick around much longer on the liver cell surface, dramatically increasing LDL clearance from the blood. These drugs are incredibly powerful, especially for patients who don't get enough response from statins alone or for those with severe FH.
It's amazing finding a rare genetic variant that protects against disease directly led to a major new class of drugs.
It's a perfect example of human genetics driving therapeutic innovation.
And we're seeing similar targeted thinking for Marfan syndrome now too, right? Moving beyond just surgery.
Yes, that's been a major shift. For decades, management was mainly monitoring the aorta and intervening surgically when it got too dilated. But the realization that excessive TGF-beta signaling is a key driver of the aortic problem opened up pharmacological possibilities.
Targeting the signaling, not just the structure.
Exactly. This led to clinical trials using drugs that were already available for other conditions, but happened to also inhibit TGF-beta signaling. specifically angiotensin II receptor blockers, or ARBs, like losartan.
Drugs normally used for blood pressure.
Yes, but they were found to have this beneficial off-target effect on TGF-beta pathways. Clinical trials, initially in mouse models and now in humans, have shown that these drugs can potentially slow down the rate of aortic root dilation in Marfan patients.
So offering a way to potentially stabilize the aorta with medication, maybe delaying or even preventing the need for high-risk surgery.
That's the hope. It's still an area of active research to figure out the optimal timing and dosage, but it represents a huge step forward using molecular understanding to target the underlying pathological process, not just its structural consequences.
And looking broadly, these examples from FH and Marfan seem to reflect a general trend in treating genetic disorders, right?
Absolutely. The core principle is understand the precise molecular defect and then devise a strategy to counteract it. Whether that's replacing a missing enzyme, like in some lysosomal storage diseases, reducing the buildup of a toxic substrate, using drugs like statins to boost a pathway, inhibiting a negative regulator like PCSK9, or maybe even using molecular chaperones to help stabilize a slightly misfolded but partially functional protein.
All these approaches stem from that deep dive into the molecular pathology.
Exactly. It validates the whole effort of painstakingly mapping these disease pathways. The payoff is rational, targeted therapies.
Okay, let's try to synthesize this. We've covered a lot of ground on autosomal dominant disorders. What are the absolute key takeaways for our listeners?
Well, I think the first thing is just the definition. ADDs only need that single mutant gene copy to manifest. That 50% risk is central. Second, remember the clinical complexity. They're often marked by huge variability in how they look, variable expressivity, and sometimes they hide completely in a carrier, incomplete penetrance.
Right, the tricky patterns.
And third, crucially, why they're dominant often boils down to the type of protein affected. It's frequently those structural proteins like collagen or fibrillin or key receptors like the LDL receptor. In these cases, either 50% function just isn't enough, haploinsufficiency, or the faulty protein actively messes things up, dominant negative effect. And sometimes it's that toxic gain of function, like in Huntington's.
That link between the protein type and the inheritance pattern seems critical. The journey we took from seeing the family patterns to understanding the molecular how haploinsufficiency, dominant, negative, gain of function, that really highlights something important, doesn't it?
Yeah, it shows that in genetics, knowing how the mutation causes damage is just as vital as knowing which gene is involved. That mechanism dictates the disease and, increasingly, the treatment.
And we've seen how medicine is now turning that how into actual therapies, statins, PCSK9 inhibitors, maybe ARBs, for Marfan ways to restore balance or stabilize things. It really makes you think, what's next? So here's our final provocative thought for you, the listener, to chew on. We focused on clear, dominant disorders. But could there be other diseases, maybe ones we currently think of as purely recessive, or even complex multifactorial diseases, where subtle dominant negative interactions are actually playing a role, where one faulty gene copy isn't silent, but is quietly sabotaging the function of the normal copy, contributing to the disease in ways we haven't recognized yet?
That's a fascinating question, uncovering those hidden interactions.
Yeah, if we could find them, it might open up completely new ways to think about therapies, using that same kind of rational design we just discussed. Something to think about.